we are going to see about the mechanism of DNA replication and what are the enzymes involved in DNA replication. So the double-stranded DNA helix and it unwind and gets separated and it forms a replication fork. This is a parental strand, they act as templates, so template strand. Both the strands they get separated and they form a fork-like structure. It's called replication fork. The various enzymes, DNA polymerase, DNA ligase, DNA uh, gyrase also we can use, polymerase 1, 2, 3 and topoisomerase are the enzymes involved in this replication. And there are two strands of DNA. Daughter strands are formed. One is continuous strand called leading strand. Another one is discontinuous strand. The break will be there. The fragments. It's called Okazaki fragments. And this fragmented new daughter strand is called lagging strand. We'll discuss all these mechanisms of DNA replication in this one by one. Now coming to the point, what are the enzymes involved in DNA replication? In prokaryotes and eukaryotes, some of the enzymes which differ. In prokaryotes, we can say DNA polymerase 1, it's commonly known as Kornberg enzyme, and DNA polymerase 2. Both are involved in DNA repair mechanism. If there is any misfault in base pairing, the repair mechanism is done by DNA polymerase 1 and 2. And DNA polymerase 3 is a main enzyme for DNA replication where the polymerization of nucleotides will occur in eukaryotes. Not for DNA polymerase 1 to 3, DNA polymerase 1, 2, 3, 4, 5 enzymes are there. And all are involved in DNA replication process. It is nothing but polymerization of nucleotides. Many nucleotides or polymers of nucleotides are formed by with the help of the enzymes DNA polymerase 1, 2, 3, 4 and 5. And what is the use of DNA ligase? It's common. These enzymes are common in prokaryotes and eukaryotes. DNA ligases, they join the DNA strands. If two fragmented Okazaki fragments are there, they are joined together by DNA ligase. Helicase is an enzyme. The double-stranded DNA gets separated or unbinding of the DNA strands with the help of the enzyme helicase. And the enzyme called topoisomerase or gyrase. This is just before unwinding, pulling apart the DNA strands with the help of topoisomerase. And nuclease is the enzyme which is involved in DNA repair mechanism. Okay. Now we'll see what are the components involved in DNA replication. Apart from the enzymes we have seen so far, the other two important components are, one is the RNA primer. In both the strand, leading strand and lagging strand, first the RNA primer with the help of the enzyme primase, it is formed as complementary daughter strand. RNA bases are added and they form RNA primer. It is RNA primer is nothing but a short stretch of RNA bases. They only initiate the mechanism of replication or initiating the polymerization. So it occurs in 5' to 3' direction. Note the direction. So the mechanism of replication always occurs in addition of nucleotides occur in 5' to 3' direction like this. Right? And the another one component is called deoxynucleotide triphosphate. This is DNTP. It provides the energy for the mechanism of DNA replication which act as substrate also. In the picture you can see the enzymes helicase. And helicase is an enzyme. It starts separating the two strands. Double stranded gets separated into single strand, and before that the topoisomerase pulling apart both the strands separately. Pulling apart means separating. And this is helicase is said to be unwinding or unzipping. Zip up open பண்ற மாதிரி. So it's called as unzipping of DNA is with the help of the enzyme called helicase. And the RNA primer which initiate the DNA replication by the addition of nucleotides, it starts only with the help of the enzyme primase. Okay, we'll discuss one by one next. Now the mechanism of DNA replication in prokaryotes, first of all we will see. in E. coli, Escherichia coli, which has 4.6 into 10 power 6 base pairs, BP na base pairs, and it completes its replication within 38 minutes. It takes its one replication of one DNA strand into two. For that, it will take 38 minutes. Here in this E. coli, the replication is very fast and accurate. Any error in the replication can be corrected. So mechanism of DNA replication or repair of DNA replication is done by the enzyme called nuclease. Here the replication, it begins at a site that is called as ORI site or origin of replication site of DNA. This is the site called origin of replication. It starts, right? In prokaryotes, they have only one origin of replication site, but in eukaryotes, they have many origin of replication sites. Okay, at this place, the enzyme called, that is origin of replication site, the enzyme called helicase and topoisomerase unwind. First here, the topoisomerase, it can attach and pull both the strand apart, right? And unwinding, it starts helicase, it attach in this place, and it result in unwinding or unzipping of the DNA strand. So it result in, what is it called? The replication fork is formed, right? Y-shaped replication fork is formed because of the enzyme helicase topoisomerase unwinding, pull apart the DNA strands and forming the Y-shaped replication fork. Now the two replication forks at each origin of site, origin of replication site. So it looks like replication fork. The two strands are uh, that is parallel to each other, already anti-parallel. Here G is there, here C. Here A is there, here D, like this, anti-parallel to each other, right? Now we'll see about the two strands which is informed. This is the parental strand. The blue color is called parental strand. It's also called as template. Okay, as a basis. And for this template or parental strand, the complementary new strands, red color is the new daughter strands which is formed. Here the two strands are formed. One is called continuous strand called leading strand. Another one is called discontinuous strand called lagging strand. So why this leading strand is called as continuous strand? Because here leading strand, the replication occurs continuously. So that's why it's called leading strand. And it occurs in, if the template strand with, this is the template strand, with the 3' and 5' polarity. To this, the replication is continuous, the new strand, leading strand is formed. And another strand, we call it as the 5' to 3' direction in polarity. Here the replication is, this is the template strand, the replication is discontinuous. Okay, that's why you can see 5' to 3' direction, the nucleotides are formed and Okazaki fragments are formed. And these fragments, Okazaki fragments, these are later joined by this yellow color enzyme called DNA ligases. Okay. So in the leading strand, replication is continuous. In the lagging strand, replication is continuous. So due to this in discontinuous replication, the Okazaki fragments are formed, but later these fragments are sealed or joined together by DNA ligases. Now, the DNA polymerase is an enzyme which binds to the replication fork and adds new nucleotides to the 3' carbon position. So, always the replication occurs in which direction? 5' to 3' direction, forming a new strand that is complementary to the parental strand. You can see the replication occurs from 5' to 3' direction, from 5' to 3' direction. So, forming the new strand which is complementary to the parental strand. So, this is a blue color is a template strand or parental strand. The red color is a daughter strand, and one of the daughter strand is leading strand because here the replication is continuous. Another new strand is called as lagging strand because this, here the replication is discontinuous but forming Okazaki fragments which are sealed by, that is the fragments are sealed or joined by DNA ligases. Addition of nucleotides which occur in the fragment fission by means of the enzyme called DNA polymerase. Okay. Always the replication occurs in 5 prime to 3 prime. Addition of nucleotides with the help of DNA polymerase, which occurs only in the direction of 5 prime to 3 prime direction. Now we'll see about the formation of new strand. It requires, at the initiation of this replication, it requires one more important one is called as RNA primer. For the initiation of replication, what is required? RNA primer. So here you can see the small portion is called as RNA primer, and this RNA primer only initiate the replication, and this is one Okazaki fragment and another Okazaki fragments, and Okazaki fragments are sealed later by DNA ligases, and this is the lagging strand. Here the Okazaki fragments are formed, and this replication starts with the RNA primer with the help of primase enzyme. And here already I said topoisomerase. What is its function, ma? It is nothing but it pulling the both the parental strands. They pulled apart with the help of the topoisomerase. Topoisomerase is an enzyme. It pull apart both the strands. They pulled apart separately, and helicase is an enzyme which helps in unwinding or unzipping of the DNA double helical strand into two separate strands. Helicase. Primase, with the help of primase DNA, that is RNA primer is also added, and the replication starts. Okazaki fragments are sealed by which one? DNA ligases. And this is continuous strand, so it's called as leading strand. And this is Okazaki fragments are there, that's why it's called as lagging strand. Is it clear, ma? So DNA replication is nothing but it is in the form of, and the gaps are sealed by DNA ligase. I said DNA replication is by means of semi-conservative DNA replication. Let us see one animation video for a DNA replication process. Okay, so DNA replication is a semi-conservative mechanism. Here we will see about DNA replication. DNA is a molecule. It is made up of two strands which are twisted around each other in a double helix shape. Okay, each strand is made up of four nitrogenous bases, chemical bases: A, C, adenine and cytosine, guanine and thymine. So the two strands are complementary to each other. In one strand, if thymine is there, in another strand, A is there, adenine. In one strand, if cytosine is there, complementary, guanine is there in another strand. So both the strands they run in 5 prime to 3 prime in one strand, and another strand is 5 prime 3 prime to 5 prime. They are parallel to each other. Okay, they run in opposite direction. Both the strands they run in opposite direction. The enzyme called first step is helicase enzyme, which causes unzipping is done by helicase enzyme. Formation of separated replication fork is formed. Okay, it results in formation of replication fork. The separated strands act as template, and the new primase enzyme is added. It starts the process of DNA replication by addition of RNA primer. RNA bases are added. This is the RNA primer. It makes a starting point for the construction of new strand of DNA. Now the third enzyme called DNA polymerase. This DNA polymerase and makes binds to the primer and makes the new strand of DNA, and DNA polymerase can add DNA bases only in which direction path? 5 prime to 3 prime direction. The addition of nucleotides will occur, DNA bases will occur. Okay, now this is called leading strand because its replication is continuous. Okay, and 5 prime to 3 prime direction occur. Another strand called lagging strand where the that is it runs in opposite direction. So here this is lagging or Okazaki fragments are formed here, and each fragment is added with the RNA primer, and with the help of the enzyme called primase, RNA primer is added, and it is followed by here Okazaki fragments, RNA primer, it is also added, and further in lagging strand DNA polymerase, which can add more nucleotides in 5 prime to 3 prime direction, and it join. Okay, and one more, the primase, once again it formed the RNA bases and primer is there, and RNA primer, it is followed by DNA polymerase enzyme at the nucleotides in 5' 3' deduction. And another DNA polymerase, it fills and the exonuclease which remove all the primers. This exonuclease remove all the primers, and DNA polymerase, it fills the gap adding nucleotides. This is DNA polymerase. Finally, the enzyme called DNA ligases. It seals the fragments and it forms continuous double strand. So, mechanism of DNA replication is called as semi-conservative mechanism. It conserves one strand from the parent, another strand, which is a new strand, is formed, and this is semi-conservative DNA replication. Thank you.